Hello everyone, my name is Christopher Ochoa. Today, I will be presenting my project titled MicroRNA Biomarkers and Genetic Factors in Chronic Kidney Disease Progression. Chronic kidney disease is a major and growing public health concern. In the United States alone, it affects about 35.5 million adults, and globally, roughly 10% of the population is impacted. The major risk factors include diabetes, hypertension, obesity, and cardiovascular disease. One of the biggest challenges is that CKD often progresses silently, without noticeable symptoms until significant kidney damage has already occurred. Because of this, early identification of disease progression remains extremely difficult. Currently, CKD is monitored using markers such as estimated GFR, serum creatinine, and albuminuria. While these tools are essential, they have limitations. Many of these changes only appear after structural kidney damage has already taken place, and they don't reliably predict who will progress more rapidly. This gap highlights the need for earlier, more sensitive biomarkers that can detect disease activity before irreversible damage occurs. Estimated glomerular filtration rate, or eGFR, is a calculation used to estimate how effectively the kidneys filter waste products from the blood. It is commonly used to classify CKD severity. As kidney disease progresses, the GFR number goes down. In this graphic, we see the stages of chronic kidney disease and their correlated eGFR numbers. MicroRNAs are small, non-coding RNA molecules that regulate gene expression. They influence key biological processes such as inflammation, fibrosis, cellular metabolism, and tissue repair. Because they respond early to cellular injury, microRNAs have strong potential as early biomarkers of kidney damage. In this project, I focus on two microRNAs, primarily miR-21 and miR-29C. miR-21 has been linked to renal fibrosis, inflammation, and declining kidney function. miR-29C is associated with extracellular matrix remodeling, fibrosis, and CKD progression. Both offer potential advantages, including non-invasive testing, earlier detection of disease activity, and more personalized risk assessment. The purpose of this project is to evaluate the evidence supporting microRNA biomarkers, particularly miR-21 and miR-29C, as tools for predicting CKD progression and improving risk stratification beyond traditional clinical markers. The research question is, in adults with chronic kidney disease, does monitoring microRNA biomarkers and genetic factors associated with inflammation and fibrosis, compared with standard clinical monitoring alone, improve prediction of kidney disease progression? This question is structured using the PICO framework, where the population is adults with CKD. The intervention is monitoring miR-21 and miR-29C along with genetic biomarkers. We are comparing with standard monitoring, which includes eGFR, creatinine, and albuminuria monitoring. And the outcome we're looking for is earlier detection and improved prediction of CKD progression. In this section, I define key terms used throughout the project. A biomarker is a measurable biological characteristic that indicates normal biological processes, disease processes, or responses to therapeutic interventions. CKD is the abnormalities of kidney structure or function present for at least 3 months with implications for health. eGFR is a calculation used to estimate how effectively the kidneys filter waste products from blood and is commonly used to classify CKD severity. We have the different stages of chronic kidney disease 1 through 5 and their eGFR limits. Some more definitions here. Albuminuria is the presence of albumin in the urine and it indicates kidney damage. We have gone over what a microRNA is, which is a small non-coding RNA molecule. MicroRNA 21 or miR21 is a microRNA associated with inflammation, fibrosis, and progression of chronic kidney disease. MicroRNA 29C, miR29C, is a member of the miR29 family that has been associated with renal fibrosis and declining kidney function. And fibrosis is long-term damage that often leads to scarring, excessive development of fibrous connective tissue as a response to injury or chronic inflammation. One of the fundamental studies by Chow and colleagues showed that miR21 promotes kidney fibrosis by suppressing metabolic pathways. This study identified a mechanistic role for miR21 in CKD progression, providing strong biological rationale for using miR21 as both a biomarker and a therapeutic target. Lenga and colleagues examined urinary exosomal miR21 in CKD patients and healthy controls. They found that miR21 levels were significantly elevated in CKD and were associated with kidney injury. This supports the idea that urinary microRNAs can serve as non-invasive biomarkers. Hanino's study demonstrated that increased renal miR21 expression is associated with fibrosis and poorer renal survival rates. This links miR21 not just to injury, but to long-term clinical outcomes. Glowacki and colleagues found that urinary miR21 correlates with interstitial fibrosis and tubular atrophy. This suggests that miR21 may reflect structural kidney damage, making it a valuable marker for early detection. In patients with diabetic kidney disease, urinary exosomal miR21 was elevated and associated with albuminuria. This demonstrates that miR21 has utility across different CKD etiologies, not just one specific cause. Liu and colleagues studied miR29C and found correlations with eGFR and tubulointerstitial fibrosis. This suggests that miR29C may serve as an additional biomarker, complementing the information provided by miR21. Now, let's focus specifically on the therapeutic targeting of miR21. Gomez et al. Showed that anti-miR21 treatment slowed the progression of kidney disease, suggesting that blocking this microRNA can positively influence disease trajectory. Similarly, Colling et al. Found that silencing miR21 improved outcomes in diabetic kidney disease. Taken together, these studies highlight something important. miR21 may function not only as a biomarker of kidney injury and fibrosis, but also as a direct therapeutic target. This dual role strengthens the case for integrating microRNA-based approaches into future CKD management strategies. Across studies, elevated miR-21 is consistently associated with fibrosis, kidney injury, and poorer renal outcomes. miR-29C is also linked to fibrosis progression. Importantly, these biomarkers may detect disease activity before substantial declines in eGFR occur, offering a window for earlier intervention. In conclusion, miR-21 is the most consistently studied microRNA in CKD and shows strong associations with fibrosis and disease progression. miR-29C also demonstrates promise as a complementary biomarker. MicroRNA biomarkers have the potential to significantly improve early detection and risk stratification in CKD. However, more large-scale clinical studies are needed before these tools can be implemented in routine practice.